Welcome, class. It's good to have you back for a science lesson this evening, a lesson that we pray will give you an outstanding view of the great God that we serve. Thank you for being with us this evening, and I have much to say, so I'm not going to do much by way of introduction. I'm going to start this way. You see the topic for tonight, the great designer, evidence from the chemistry of living things. You live in one of the most exciting times in the history of chemistry because we've learned more about the chemistry of living things in the last 100 years than in all of time before that. And it's an exciting time. Let me give you some illustrations of what we've learned in the last 100 years. It was in the 1950s that Crick and Watson discovered the structure of DNA. If you were alive then, raise your hand. 1953. Okay, you make me feel better. I was 8 years old, but it was exciting. That was a breakthrough. Crick and Watson won the Nobel Prize for that some years later. What you probably don't know is Frederick Sanger discovered the structure of insulin in the 1950s also. That was the first protein that was mapped. He won the Nobel Prize for that in 1958. And Sanger is one of the very few people in the history of mankind that won the chemistry award and the Nobel Prize twice, because he also mapped the DNA sequence in 1977. The space program. Class, you're allowed to talk. This is my class. What do you remember from the space program in the 1950s? Thank you. Say that again. Sputnik. You youngsters know what Sputnik is? For those of us that were alive then, it scared us to death. Because who put Sputnik up in space? The Russians did. We knew they were going to take over space and rule the world, which that's what they wanted to do anyway. So, our nation reacted to Sputnik. A whole lot of money went into science education because we were clearly behind the Russians and we needed to improve science and math. And out of that came a whole set of curriculum studies to rewrite the curricula for elementary, middle school, and high school, and college in all those subjects. And if you'll pardon me to take a little side trip here, I was a partial beneficiary of that. Because one of the things our federal government did was they gave Harvard University a bunch of money to make better science teachers. And they gave out that year, 1967 is the year they got it, is they gave out 50 scholarships, full rides to get a master's degree in science education. I happened to get one of those, folks. And I was privileged to be a part of some of the rewriting of chemistry curricula. It was called Chem Study. And about the same time, there was the new math. You parents remember the new math? Parents hated it because they didn't understand it either. But the fact is, we made a lot of moves in the 50s and 60s to improve science education. You probably remember that we beat the Russians to the moon. And it was 19 what, class? When we went 1969, when we put a man on the moon. Dramatic. So I'm saying to you, you live in one of the most exciting times in the history of chemistry and science in terms of living things. April the 8th, 1966. This was the headline article in Time magazine. Is God dead? Now, I've said to you before that one of the things that was happening as we discovered more and more about how life works is that people began thinking in the scientific realm especially, that means we don't need God for that because we've understood now how it works. And we're going to eventually be learning so much that we don't need God anywhere. And that was happening. And it was happening in the educational world, especially in the sciences, as teachers were telling students, look at everything we're learning here. And I was one of the recipients of that, which is part of the reason I'm doing what I'm doing right now. Because as a young person, I was impacted by this. Didn't know how to answer these things. It was very disturbing. Well, it's interesting that about that same time, a gentleman named A.W. Dicus wrote the song you just sang. Our God, He is alive. It was an answer to this question. A.W. Dicus was the academic dean at Florida College during those days, and I was privileged later to literally sit in his seat because I served as dean at Florida College for 18 years. He was a physicist and a well-respected scientist, and he openly declared his faith. And you remember the third verse of that song that we just sang? Secure is life from mortal mind. God holds the germ within his hand. Though men may search, they cannot find, for God alone will understand. I'm quoting that, and I had them sing it tonight because we had a professor in biology at Florida College in those days who refused to sing that verse because he didn't believe it was true. He was in a doctoral program at Florida State University in biology, and he was convinced that man was going to know how life came about, and it may not be just God that knows. So he wouldn't sing verse 3. I've sung verse 3 all along, folks. And I wish he were alive today, because in 50 years since then, we have learned so much more about life and how complex and amazing it is. We know less, and we don't have a clue today how life first got started without God. There is no answer to that. In fact, we're farther away from it than we were then, by far. And that's part of what I want to explain to you tonight. So welcome. In this article and in the magazine, Thomas Altizer and William Hamilton, two theologians class, in their book Radical Theology and the Death of God, said this: we must realize the death of God is an historical event, that God has died in our cosmos, in our history. The dead God is the God of the historic Christian church and beyond the church of Christendom at large. So while in the scientific community, you were having science teachers say, we're going to learn how life came about and we're going to learn how to do it ourselves. You don't have to have God for that. The theologians in some churches were teaching their parishioners, God died. How in the world is that? That God died. The only way God could die is if we made him up in the first place. And that's what I'm afraid a lot of folks believed. So I'm telling you, ladies and gentlemen, this is a real live subject that needs to be talked about. And when I was in college, it was a heavy subject being dealt with by youngsters who didn't know how to answer those questions. I like this book better. Is Atheism Dead? It's by Eric Metaxas. He's not a scientist, but he's a well-read gentleman and a good writer. And in this book, he argues that in the 2000s, there's better evidence than ever that atheism should be dead. So here's one of the statements he made, and I like it a lot. Since the Time article in 1966, because this is a takeoff from it, roughly 5 things have arisen to challenge, and I will argue to overturn, the secular consensus that formed in the wake of that article. Can I explain that to you briefly? That Is God Dead article in 1966 had an impact on our society. There was a secular consensus that flowed out of that said we don't need God. And it was very broad. And we're still, reaping the results of some of that. He's arguing that what we've learned in this century, the last century and a half, is now going to overthrow that consensus. So here he says, there are 5 things that have happened in this century, starting with the 20th and into the 21st, that overthrow that consensus that was had then. First, we've learned that there's a beginning. Did we talk about that last night briefly? It's not the theme of my subject this week, but it certainly is well documented. The evidence from science is there was a beginning. Please go look it up if you have deeper interest in that one. Second is fine-tuning. Everywhere you look, it sure looks like this earth and all the things surrounding it are fine-tuned so that you can be here. And if it weren't fine-tuned, you wouldn't be here. The evidence of science is overwhelming in that regard. Did we talk about that last night, class? And you remember our favorite topic that I made you say over and over again? Everything has to be, or you wouldn't be here. And that is the truth. The fine-tuning argument is not to be refuted. It's too heavy. There's evidence everywhere that fine-tuning is a fact of life. Powerful evidence that somebody tampered with this. How about designed it? It didn't come about by natural causes. The third thing he mentions is stupefying complexity. I love that expression because I want you walking out of here tonight stupefied about the complexity of living things. So my job is to stupefy you tonight. And it's not that hard if you'll really listen. But I am going to have to give you some detail. Please understand, there will be a test tomorrow. No, I'm kidding. I'm not expecting you to have to remember all of this. What I'm expecting is for you to be overwhelmed with the majesty of it and the wonder of it. The fourth thing he mentions is archaeological discoveries that support the text of the Bible. That also is not my topic tonight, but that's the case also. And fifthly, we have learned in the last century what atheism is and what it produces. I'd like to take some time tonight to read you some of the things he says in this book about the 20th century. It's the century of atheism. Governments run by people who did not believe in God. The Nazis and the communists killed more people than any time in the history of mankind. That's what communism and Marxism and Nazism has given us. And they did it on the basis of their belief system, which did not include God. You want to know the results of atheism? You look at the 20th and 21st centuries, and you will say, with me, I hope, atheism is the last thing we need. It needs to die. But that's a topic we could discuss at some length, but that's, again, not the topic for tonight. We're going to focus in on stupefaction. So, welcome, class. I told you last night that when I do this kind of a lecture in a church building with lots of believers here, I always want to start with the Bible. So we're going to turn again to Romans chapter 1. Would you please? Verse 20. It's the theme passage from the Bible. And why do I do that? I want you to know, Bible believers, of which I am one, that the Bible teaches you can learn there's a God without the Bible. Did you hear me? The Bible teaches you can learn there's a God without the Bible. Romans 1:20. Since the creation of the world, His, that is God's, invisible attributes are clearly seen, being understood by the things that are made, even His eternal power and Godhead, so that they are without excuse. That passage teaches you can know there's a God that is eternally powerful and that is not part of nature. He's outside of nature. From examining the things that are made. It's the famous and powerful argument from design. So the Bible teaches that it's perfectly legitimate to be doing what we're doing tonight. So, as I said last night, for the rest of my time tonight, I'm closing this book. Not because I don't want to preach from the Bible, I do. But God has another book. And that's the book we're going to look at together again this evening. So welcome, class. Whether you meant to or not, you're in class 1 of biochemistry. Welcome to day 1. So happy to have you in my class. I told you last night I have a few rules. One is I don't allow sleeping. And I didn't tell you what I would do if you start sleeping, but here's what I'm going to do. If you start sleeping, we're going to pause and everybody's going to stand up and shake yourself. And why do I do that? Because you're going to miss something I'm saying. And if you do, you won't be as stupefied as I wanted you to be. Because I don't want you to miss anything. And look, I understand. If I were sitting where you are, if I had a nice supper tonight, I'd be going to sleep myself. So I understand the problem, but I don't allow it. The second thing is, I really want you to participate. I got you to do that pretty well last night. Doesn't that help keep you awake? I think so, and alert. So I really like participation, not picking on anybody, but try to keep you alive and well. So here we go. Chemistry of living things. That's what biochemistry is about. The chemistry of living things. I love teaching it. If you didn't like chemistry, it's because you didn't have me. I'm joking. But I am enthusiastic about the subject. And had you had my class, you too would become enthusiastic, or you wouldn't have made as good a grade as you wanted to. The critical components of life, class. This is the chemistry of life. So we're going to start by studying how is life composed. I thought to do that, we might ought to use a book by one of the most famous authors of our time named Francis Crick. If you were listening to me at the beginning of my introduction, what do you know about Francis Crick? The co-discoverer of the structure of DNA. Francis Crick. He ought to know a lot about life, shouldn't he? One of the Nobel Prize-winning chemists. So he wrote a little book in the early 1980s called Life Itself, Its Origin and Nature. So this book is supposed to tell you what life's all about and where it came from in the 1980s. So I'm going to read a little bit from this book. And while we'll do it, we'll lay out the chart here before you. You see on the screen. What are the critical components of life? Here's what he says. Life as we know it on Earth appears as a synthesis of two macromolecular systems. And I just lost you, didn't I? Let me put it in plain English. Every living thing has two kinds of giant molecules. That's what he said. Two kinds. The proteins, because of their versatility and chemical reactivity, do all the work, but are unable to replicate themselves in any simple way. So the first kind of molecules, class, that's in every living thing is proteins. So would you please say that for me? Good. That, class, will be the answer to most of the questions I'm going to ask you tonight. So you got it? Okay, proteins. Extremely important. That's one class of molecules that are in every living thing. I think you got that. That's question one on the quiz. No. The nucleic acids seem tailor-made for reproduction, but can achieve rather little else compared with the more elaborate and better equipped proteins. RNA and DNA are the dumb blondes of the biomolecular world. Fit mainly for reproduction with a little help from the proteins, but of little use for much really demanding work. I didn't say that. Don't you get on me for that. He said that. But it's a way to remember it. What do the nucleic acids do? They're primarily for reproduction. But they don't do a lot of the work. The proteins do a lot of the work. But class, every living thing has both of those. Proteins and nucleic acids. And by the way, listen to me closely. Every living thing is made up of the same chemistry. The proteins and the nucleic acids, they're made of the same stuff. If you're a one-celled bacteria or if you're a human being, that's the truth. Now he goes on to say in this paragraph, the problem of the origin of life How did life get started? Would be a much easier task to approach if there were only one family of giant molecules capable of doing both jobs, work and reproduction. But life as we know it always employs two. And I'll say again, you have to have both or you don't have life. This may well be due to the fact that no macromolecule exists that could conveniently carry out both functions. I'll tell you what we know now, class. We know good and well no one set of molecules could do both. But you got to have it to have life. So I think what I'm going to do is skip over just a little bit to page 88. For I may forget this later. An honest man, I'm quoting Francis Crick, an honest man armed with all the knowledge available to us now, and this is 1980s. We've learned a lot since the 1980s, class. And the picture's a whole lot worse for Francis Crick and his kind today than it was then. An honest man armed with all the knowledge available to us now could only state that in some sense the origin of life appears at the moment to be almost a miracle. I'm fine with that, with everything except the word almost. So many are the conditions which would have had to have been satisfied to get it going. He's saying that in the 1980s, class. In 2026, he would ought to multiply that by 10 billion. It's way more complicated than we had a clue about in 1980s. The plain fact is that, now he goes on to try to explain it away. The plain fact is that the time available was too long, the many microenvironments on Earth too diverse, the various chemical possibilities too numerous, and our own knowledge and imagination too feeble to allow us to unravel exactly how it might or might not have happened. In other words, we don't have a clue yet, but we can't give it up. And you know what he does after this, class? If it was highly likely that life just started by itself, then there's no problem. Well, that's got to be the understatement of the century. But if it turns out that it was rather unlikely that life started all by itself, then we're compelled to consider whether it might have arisen in other places in the universe where possibly for one reason or another conditions were more favorable. And you know what he does with the rest of this book, this Nobel Prize-winning chemist? He discusses directed panspermia. You have any idea what that is? Directed means somebody sent it. Panspermia means something that was alive that was sent here. So he's taken the position in this book that somebody sent the first living forms here on a spaceship. He has a whole chapter of what the spaceship might have been like. Class, is there one scintilla of scientific evidence that some civilization out there sent the first life forms here on a spaceship? Well, you know, they're talking about it again right now. There isn't a scintilla of evidence that's supported well that that's the case. But here's the Nobel Prize laureate. But he's not going to let God in the door. What I've learned from these guys is they will take anything but God. But I can't even imagine this Nobel Prize winner writing a book about life itself and spending half of it on something we know nothing about. Just a made-up story. So let's look at a summary here. Life has structural materials. Those are proteins, class. And they're assembled from amino acids. And they make up the shape and mobility of every living thing. Second, there are tools and machinery, we call them enzymes. If you're not familiar with that, an enzyme is a special case of a protein. Its job is primarily to speed up or slow down things. And they're very important. And they make up the growth and metabolism, the maintenance of living things. So did you see that both of those are proteins? And they do all the work. But the third great class is blueprints. They're the genes, the nucleic acids, it's the RNA and the DNA, and they're primarily for reproduction. They contain information and directive function. All living things are like that. Proteins, class, are made up of, as you see there up there, from amino acids. In day one of biochemistry, that's the first thing we're going to talk about. There you are. I'll pause a minute and let you memorize that. You got it? You ready to move on? Class, what is fascinating is that those 20 common amino acids occur in all proteins in all living things. Does that bother you that you've got the same amino acids as E. coli? You do. And the proteins in that little bacteria are made of the same stuff as your proteins, these amino acids. Okay? So let me give you my first simplified definition of a protein. It's a string of amino acids. So class, when I say to you a string of amino acids is that's what you're going to say. We may say that 10 times before this class is over because repetition is the key to knowledge, and that's the one thing you'll remember when you walk out of here. A protein is a string of amino acids, class, and it'll be one of those 20 in the string. There's a whole string of them. Let's go through it just a little bit. Why are they called amino acids? A little bit of chemistry here. Do you see that little NH3 group on each one of them? There's an NH3. NH3 is an amine group. It's a nitrogen and 3 hydrogens. So they call it amino. Every one of them has an amine group. Second, why is it called an acid? Because you see that COO group right there and there and there? Every one of them, these has a COO group. That's a carbon attached to 2 oxygens. And you notice, if you will, that at the end of it, there's a little minus. Can you see that? A little negative. Because it's dropped off its hydrogen ion, and that's what makes you have an acidic solution. So a COO group has produced acid. Therefore, it's called an organic acid. So it's an amino acid. That's question number 2 on your quiz tomorrow. And the reason that's important, class, is because that's in every one of these. It's a unifying principle. You'll also note that after that, they're all different. Each one has its own unique properties. They share those 2 common things, but other than that, they have their own unique properties, which means, class, they have their own unique chemistry, which means they do different things. And they therefore help your body do different things. Okay, I think you're with me. Now, let me ask you this. How do you think an evolutionist explains that every living thing has the same chemistry? Anybody want to take a jab at that? How do you think an evolutionist would explain that? Who believes there's no God? It all came about by natural causes. Well, I'll tell you how. Every living thing came from the same common ancestry. If you're an E. coli, you're in the same tree of life with a human. Humans come from up here. We trace our ancestry back far enough. We all have a common ancestry. The first simplest life forms were one-celled organisms which had this chemistry, and all the rest of us came from that, so the chemistry is the same. That is not a stupid answer, class. That is a possible answer to this question. And it's not unreasonable, that answer. We have to look a whole lot farther than that to see which is the better answer. What does a creationist say? If God were going to make a creation that was interplaying and that fed on itself, you have to have a system by which they can feed on one another and grow and develop. Why would he have to create new chemistry every time he created something? It makes great sense to have a common chemistry in a living system like we have, made by a designer. So that's certainly as good an answer as they give. But there are two answers to that question. So we must go further, folks, to really get a solid answer. But it is the case, please don't deny facts. The facts are living things have proteins made of this. Period. I'm pausing to kind of let that settle in. Every protein is a combination of these 20. It's very much like the alphabet in English. There's 26 letters in the English alphabet. And isn't it the case, class, that every word in English is some combination of those 26 letters? By the way, it sure seems to me like that alphabet is designed. I don't believe there's ever an instance of anything we know about where you have an alphabet with a language that didn't come from an intelligent source. That's just thrown in free of charge right there. But it is a fact that amino acids, these 20, make strings, and when you get a string so long, that's a protein. That's what they are. All right. That's not nearly enough time for proteins. If you were in my class for real, we'd spend the first 6 weeks on nothing but proteins. I have to move on. DNA and RNA are the nucleic acids, and they're extremely important because they're involved with reproduction. And they have properties. One of them is this amazing double helix structure that Crick and Watson discovered. And kids, y'all have studied this, haven't you? Most everybody studied this these days. This is no news to you all. How about you old-timers? Have all of you studied this somewhere? Maybe a little bit. You've at least talked about it because you've heard it talked about in court cases, haven't you? Because people use DNA to convict a person of crime now. Because DNA can be unique to your system. They have different features. But it's double helix with crossbars. Let me see if I can see that strand right there comes wrapping around, and here's another one wrapped around it. And you see it has crossbars. And the way this color is diagnosed is there's only 4 colors. And notice that if there's a blue one over here, there's always a green one across from it. And if there's a red one over here, it's yellow across from it. That's because there are 4 bases that form the cross pieces, and only certain ones go together. Adenine, thymine, guanine, and cytosine. What do you think is question 3 on your test tomorrow? Those 4 bases, A, T, G, and C. A goes with T, and G goes with C, like a horse and a marriage and love and carriage. Horse and carriage and love and marriage. 4 bases along the strand which give you the cross pieces. I think I have that up here. That gives it the capacity to reproduce itself. Class, DNA contains a language with 4 letters. A, T, G, and C. I've already said it once, so I'll say it again. We know nothing in science or the history of mankind that has a language in it that did not come from an intelligent source. So if this were a court case and I was trying to convince you, ladies and gentlemen of the jury, that living things were designed by an intelligent being, I would say case closed. Because we have absolute proof that there's a language inside every cell in your body and every cell of every living thing. It's in DNA. And not only do we know it has a language, we know how to read it. And we know what it's doing. And it's doing amazing things. And it's based on a language, class. So I would say, ladies and gentlemen of the jury, can you believe that that kind of a system came about by natural causes, by the forces of nature? Or as Richard Dawkins would say, the blind forces of physics? I cannot believe that. I don't have that much faith. So that to me is sufficient to help folks see you should believe in God, not naturalistic science, as the source for living things. But there's a whole lot more than that. You see, we know how to read this language. It's called the genetic code. How many of you are familiar with that? You've heard it at least. So, class, what I'm needing to do for the next few minutes is teach you how to read. So look up at this table, because summarized in this table are all the possible words in this language. Because in this language, words only have 3 letters. Wouldn't it be nice, kids, if every word was only 3 letters? That'd be an easy language. It wouldn't necessarily be nice. You wouldn't have so much flexibility, but that's the way it is in the genetic code. So look, if you take the first letter over here, T, C, A, and G are your 4 letters. The second letter would be up here, and the third letter's down here repeated 4 times. You can write every possible letter that's 3 letters long if you get to repeat letters. So they're all up there. In fact, if you were in my class, we'd do the math. If you have 4 letters, you can use them 3 at a time with repeats. How many words can you make? Here's the answer. You can do 4 letters in any position. So in position 1, you can put 4 different letters. In position 2, you can put 4 different letters. In position 3, you can put 4 different letters. That's 4 times 4 times 4. Do the math. 4 times 4 is 16. Times 4 is 64. There are exactly 64 words up there. That's how many words are in this language. And every one of them is made up of 3 of those letters. So let me read for you, may I? Here's the first one. T, T, T. That's t-t-t. It's pronounced t-t-t. This is not a spoken language, ladies and gentlemen, but it's a real language. And do you notice that by the T-T-T there's a parenthesis P-H-E? That doesn't help you a bit, does it? But I will tell you that t-t-t means fee. Okay, I'm pausing. Let's try another one. You bombed that one. Let's try another one. How about C-A-T? Cat, right there. That's at least a word you've heard in another language. It doesn't mean the same thing in this language, sorry. But it does mean H-I-S, which doesn't help you either, does it? Say it again. Cat food. I can't hear you. Oh, cat's his cat. That'd be one way to interpret it. Let's try one more. How about A-C-T? That's this one, act. That's T-H-R. I'm not being very helpful to you. But see, if you had listened closely when I showed you all the amino acids, they have abbreviations. And PHE is phenylalanine. That's one of the 20 amino acids. HIS is histidine. That's one of the 20 amino acids. What did I have? ACT is threonine. That's another amino acid. So class, what you have up here is a code for all 20 amino acids with repeats. Because you only have 20 amino acids and you got 64 words. So there are repeats. I call that a backup system. There's several codes for some of these. But fact is, class, this language codes for amino acids. So not only is it a language, the words mean something. And they tie right in with reproduction. Amazing. Interesting also, I'm going to back up one. Let's get rid of that. Do you see this one right here? TAA. Can you read that? What does that say, class? Term. You have any idea what that means? How about terminator? Terminator. There are 3 codes that mean term. What that means, class, is stop. So if you read TAA, it means stop, whatever you're doing. Well, what are you doing? You're building strings of amino acids. And when you get to that code, it means stop building. You're done. It's a terminator code. I will also tell you that this one right here, where are we? ATG, which is methionine. also means start reading. So everybody's eyes on me, very carefully. You're reading along the DNA strand. DNA is a spiral. You unzip it. You read along one side of it, and you're reading a series of amino acids, aren't you? And they're 3-letter words. So you're reading along that code, and you come to one that says ATG, and it says start reading. So that's the first amino acid, methionine. And then you keep reading, 3-letter words, 3-letter words, 3-letter words, 3-letter words, 3-letter words. You get to TA, and it says stop reading. So you started reading, and you stopped reading, and in between there's a string of amino acids. What is that, class? That was weak, but that's correct. You have just built a protein because you made a string of amino acids using that code right there. And class, if you don't remember anything else, you will remember, I hope, that proteins are built by stringing along amino acids. And how's that done? It's done with a language in every cell, in every living thing that is used at an enormous rate to build what? Weak proteins, among other things. That's exhibit number 2 in my case for you. Somebody designed this. This did not come about by natural causes. Since when does a language just come about in useful form, producing incredible things without a designer, an engineer? Doesn't happen. Let's use common sense. All right. But there's a whole lot more to this story than that. Whole lot more. All we've talked about is just the process that you can actually make a protein. So, class, eyes up here. I'm watching you. Some of you are dozing a little bit. We're getting close. Transcription. In the cells in your body, humans, you have eukaryotic cells. That means they have nuclei. You see the wall here? That's a nuclei wall, nucleus wall. Most of the DNA is trapped inside the nucleus in your cells. So there it is. Do you see that it's all wrapped around each other up here? And do you see that there's a part that's unzipped, come apart? And it's exposed, both sides. And notice that one side is being copied. Do you see that? 3 different times over here, but they're copying things off of it. That's called transcription. The information on the DNA is being copied. That's what has to happen first. So what I'm about to tell you is the story of how you build one protein. You got it? I'm telling you the story of how you build one protein. Okay, I'm convinced. Everybody stand up, please. I'm losing you. Shake your body enough to get the blood flowing. I would lead you in jumping jacks, but it's not appropriate. Come on now. And no fair leaving. All right, let's be seated. Thank you. I just had to get your blood flowing again. Transcription, class, is the process of copying the information off of the DNA. And what you do when you do that is you make what's called messenger RNA. Everybody in this room should have heard of that because that was the vaccine they gave us for COVID. It was built around messenger RNA. And if I may pontificate for just a moment, one of the reasons I objected to it myself is because messenger RNA is messing with how nature produces things all the time. It wasn't like the other vaccines, which are part of the thing that caused disease that they kill and then put part of it in you. That's different. So I felt like there wasn't enough attention given to this brand new thing before they forced everybody to take it. That's Miss Mann's opinion. And again, it's because they're using that stuff in a vaccine that's never been done before till this generation. Okay. Messenger RNA is a copy of what's on the DNA. It's called transcription. Once that copy is made, it has to be released into the nuclear protoplasm. Then it has to be matured. Do you see this right here? That's got some white spots in it, and then it's matured and the white spots are gone. Those white spots are called introns. And what we've learned in the last 20 or 30 years is those introns are snipped out and then it's edited back together. That's called maturing. So you've matured the messenger RNA and the transfer RNA and the ribosomal RNA. They're all being matured, and then they migrate. That's the next thing. You see that right there? They have to move from here outside the nucleus. You say, well, that's no big deal. You got to move. Folks, that's not easy. Now let me back up a step. When you make that copy right there, the DNA has to unzip. What do you think, class, it takes to help DNA unzip? I've told you the answer. Protein. That was weak, but let me try again. What do you think it takes to unzip DNA? Protein. It not takes one protein, it takes multiple proteins to make it unzip. All right? It also takes multiple proteins, class, to zip it back up. Have you ever gotten your zipper stuck? Built into your system is a special protein that keeps DNA from getting messed up when it zips back up. Once that messenger RNA is made, you have to mature it. Guess what it takes to mature messenger RNA? Right? And then it has to migrate. Guess what it takes to help it migrate? Then it comes down here to this hole in the nuclear wall. That's a pore. Do you think that hole's going to let just anybody go through there? Not at all. Guess what it takes to open and close that pore? Guess how many things are on the walls of that pore? Proteins. There's proteins all involved here. Then once it comes outside, it's got to have help to get from here to these two ducks hugging each other. Doesn't that look like two ducks hugging each other? Class, that's a picture. That is a ribosome. It doesn't look like that at all. That's a silly picture. But it does close in on the messenger RNA like that. And it has to have help to get from here down to that so the ribosome can grab it. What do you think that takes, class? That's exactly right. And then once it gets here, this ribosome starts reading the information on the messenger RNA, which it received up here by copying it, and it has to translate it so that you can build the protein. What do you think it takes to do all that? Absolutely. And class, once the protein is constructed, which is a string of amino acids, isn't it? You think it's done? Absolutely not. A string of amino acids is useless until you do a bunch of other stuff with it. That's called post-translational activity, PTA. What do you think it does? It's required for all that post-translational activity. Exactly right. Every step of this process of building one protein requires proteins. Now, don't you miss this point. Every one of those proteins had to be built the same way. You following me? So, for example, there's a protein called RNA polymerase that allows the messenger RNA to copy the information and unzip up here. It's called RNA polymerase. That's an enzyme. That's a protein. That protein has to be built. But you got to have it to build it. You didn't get it. Every protein that I just mentioned a while ago, when you said protein 16 times, every one of those has to be built. And the very first step is to unzip DNA, and that requires RNA polymerase, which should have to be built already itself. And all of those proteins already had to be there. This is the problem. That's why Dr. Crick said maybe it happened somewhere else, because it couldn't have happened here. It's incredibly, well, you can't do it, class. It's the old chicken and the egg problem. You have to have it all present or you will not ever get anything. There's no question about this. But I haven't stupefied you yet. So we got to go through this again. In fact, I'm going through it 2 more times. But you've gotten so far that transcription requires what, class? Migration requires, translation requires, post-translation requires, everything requires, because they do all the work. And they all are built by your cells by this same process. So they all have to be there, or you ain't getting one. So there's a little blow up to make it a little clearer. I'm not going to spend time on that. We've already talked it through. So let's go back to this now. Here's what I want to hit you between the eyes with. I don't remember how many days. I have a 7-page document here. If anybody wants a copy of it, you ask me and I'll give you a copy of it. The heading of this article is the number of proteins needed to make one protein. I'm nervous about AI. I'd never used it before. But I needed so many calculations this time, I decided to let AI do the calculating for me. So I went through and asked question after question after question. I said, how many proteins does this take? And AI did the research and came back with the answer. So every bit of this came from the AI who was researching for me when I asked it questions. So I told you I'm going to give you some detail. I want to read you some of this. I'm not expecting you to remember this, but I do want you to get the point. To determine the exact number of proteins needed to transcribe a specific messenger RNA from the DNA, and all we're talking about messenger RNA right now. There's a bunch of other things that have to copy also. Consider the following points. This is what AI said. Consider the following points. Determine the number of transcription factors required for initiation. How many proteins do you have to get this started? To initiate transcription of a gene into messenger RNA, several transcription factors are required. Core promoter elements, at least 2. General transcription factors, at least 5. Additional specific transcription factors may be needed, at least 2. Enhancers and silencers, at least 2. That adds up to 11 specific proteins needed just to start it. You with me? That's part 1. Account for RNA polymerase enzymes involved in the transcription process. Did I mention RNA polymerase already to you? Yes, I did. To determine the number of RNA polymerase enzymes needed for transcription, consider the following points, says AI. Single RNA polymerase, typically 1 per enzyme is what you need, but sometimes you need 2. So there's 2 of those. We're going to count 2. That's polymerase. We're just getting started. Include co-activators and co-repressors that may influence transcription. Did you know that you have some enzymes that keep it from doing that, and they have some that make it do it faster? So they're activators and they're repressors. You need 2 of each of those. So that's 4 more proteins. I'm skipping a bunch of reading here. Consider the presence of additional regulatory proteins that may bind to enhancers or silencers. That's another day-long lecture, folks. You need at least 5 of those. I'm skipping a bunch of stuff. To understand the number of proteins needed to produce a mature messenger RNA, you remember that after it's copied, you have to mature it. How do you do that? Yeah, that's right. Here's the list. You have to have capping enzymes. You have to cap off one end so it doesn't get destroyed. You have to splice it. You know what that means? You got to cut out the stuff you don't want. And by the way, can I pause there just a minute? You know, when we started discovering that you had to cut out parts of this, and they're called introns, You know what the evolutionists said about that? They said, that's junk. It's stuff cut out and it's just thrown away. Why would God make something where you have to copy it and then you have to cut out a bunch of it and throw it away? That's stupid. So they began to call those introns and a bunch of other things they discovered in the Human Genome Project junk DNA. Did you ever hear that expression? Starting in the late 1990s and into the 2000s, Richard Dawkins loved that so much. He said the only thing that this is good for is to confuse creationists, because God wouldn't do something like that. You know what we found out, class, in the last 20 years? That junk is doing all kinds of things they knew nothing about then. You better be careful what you call junk. And what I wish some of these guys would do sometimes is be humble enough to say, we don't know what's going on. Give us about 20 years. Because in my experience, that's what's happened. Class, we now know those introns code for a bunch of little tiny RNAs that are doing all kinds of things in your body that you'd be dead if we weren't doing it. And they called them junk. My point here is, what does it require to snip them out? Say it. Proteins. A bunch of them. It mentions polypolymerase, cleavage and polyadenylation, cleavage stimulation, cleavage factors, poly-A bindings, RNA helicase. 10 different kinds of proteins, plus export proteins. Once you get it snipped and matured, and by the way, I'm coming back to that again in just a minute, it's got to move, migration. How do you transport a mature messenger RNA from the nucleus to the cytoplasm? You need nuclear export receptors. You need RNA binding proteins. You need at least 3 additional, and then when you get to that pore, you remember I told you about the hole? That's called a nuclear pore complex. How many of those? 30 different proteins in that hole are required to allow that messenger RNA in and out. That's 30 more. What about once you're outside the hole and you're going to come down here to translate? I'm skipping 2 pages worth of stuff. The bottom line is, for that whole process, it requires 1,854 different proteins. So at the end of my paper, I have a grand total. So here we go. Let me think. I'm going to do something else first. Watch this. You have to have a spliceosome class to splice and edit that, to mature that messenger RNA. I told you I was coming back to that. The spliceosome class is one of the most complicated machines at the biochemical level in the universe. Tomorrow night, my subject is molecular machines. This is one of them. What's it for? It's to take one of these messenger RNAs, and you see this part here that's a different color, that's called the intron. It's got to go, and we've got to put this one back together with that one. So all of this stuff in purple here is the spliceosome. So there's a whole section in this paper on spliceosomes. There are over 200 different proteins in that one machine. And it all works together like a charm. And every one of them had to be built by the same process. So transcription, 232 different proteins. Migration, 41 different proteins. Translation, 1,854 different proteins. That adds up to class 2,127 different proteins to build one protein. Every one of those has to be built the same way. So they all had to be there. And by the way, I took the low number if they had a range. That was my second time through this. My third time is going to be thanks to Drew Berry. Drew Berry is a biomedical animator. His love for life is producing videos that show you what's happening at the biochemical level. If you want to have some fun, go to his website. He's got all kinds of fascinating videos. I'm going to show you one. And I have the best AV guy on earth back here helping me. Right, Jacob? All right, so his job, class, is to stop and start at my command, and it has to be perfect or he's going to mess up my whole talk. No pressure, Jacob. No, I want to tell you, he's been practicing and helping me. And there's a lot of work put into this, folks. Please remember Jacob in your prayers and thank him a lot because he's really been good. And so has the whole team back there because you got sound guys and, I mean, you almost have an engineering degree to help. All right. This is a video to show you how a protein is built. Okay? Has anybody seen this before? You have. By the way, let me talk about Ronnie a little bit. When I got started doing this, Ronnie Milliner helped me make my first slides, he and his daughter. That's many years ago. But I never forget that, Ronnie. It was great help. All right, so here we go, class. We're going to talk about this. Let me highlight a couple of things here. That strand is DNA. See it? See how it's wrapped around itself? What do you think all these are, class? You know the answer. Proteins. Do you see the different colors? They are not different colors. He just does that to make it look pretty. And so you'll know they're different proteins. So here's one in red. Here's a light blue. Here's a darker blue. Here's a green. Here's a different green. Here's a different color. That's all different proteins. All of those are needed just to get started. I told you that. Now let's watch it. And one of the things that people have done with videos in the past is they've tried to make this happen, but it's never up to real-time speed. So part of what this man has done is try to not slow it down, but speed it up. So we'll see. Let's go now. Here's what happens. Here's your protein blob. And you're going to watch in a minute. You're going to see that vitamin D right there? Stop right there. Whoops, we lost it. Keep going just a little bit, Jacob. Right there. That's the vitamin D. Do you notice it's coming up to this protein? So watch. Keep going, Jacob. Boom. Stop. Did you see how it turned green? It does not turn green. Green means go. So that's why he does that. Look, class, you need vitamin A to activate this process. You think you need to be getting your vitamin A? I hope so. Because you activate that protein, which starts the process, and then you move on. All right, let's go. Gene activation by vitamin A. All right, here we go. Here's another, what do you think, class? Yep, in fact, there's more than one there. Stop. That was about 3 microseconds, and a bunch of stuff's happened there, which I don't have time to tell you. But just know that every one of these proteins has a specific job that it is doing while this starts. A specific task. Didn't have it, wouldn't happen. One of them, ribonuclease, which I've mentioned 6 times now, actually rides along the DNA. That's the big old what? Protein that rides along here and unzips it and allows it to make copies. And you see this yellow thing coming out here? That's the RNA that's being made that comes out the top. Okay, let's go, Jacob. Here we go. We're going to ride along the DNA. It's gene transcription. Stop right there. It's the transcription of the genetic code from DNA to RNA. That's what you're watching. Keep going. This is the DNA, and that's the RNA coming out the top. And now we're going to close in on this glob of what class? Protein. And we're going to look down inside it here in just a second. Right there. Stop. This is the glob of ribosome. I mean, pardon me. This is ribosome. No, I'm messing me up. RNA. I can't say it now. Ribonuclease. Pardon me. That's the glob of protein. Look at this stuff out here that's floating around in the nucleus. You have to bring it inside, once the DNA is opened up, these things are part of what build the messenger RNA. So let's watch. Go. Jacob, are you home? Okay, there we go. Now, can you see it's unzipped? Stop right there. Class, you see the code down here? GGTAAG. That's the code. Do you think those letters are written down there on the DNA? No. That's there for you to recognize. That's part of that's guanine, that's thymine, that's adenine, you see? And whatever's down here, the messenger RNA will have its mirror image. The A will always have a U, which is T. The T will always have an A. The G will have a C. The C will have a G. So what you're doing is you're making exact mirror image copy of the code on the DNA. Watch what's happening. Go, Jacob. Can you see one at a time they're being built? One. Stop there just a second, Jacob. Every one of those has to be copied one at a time. Do you know, class, that the average protein sometimes has 1,000 amino acids in a row? The average. So when you copy it, you're copying 1,000 different amino acid codes, at least. Go. Now he wants to show you how fast it happens. That's real-time speed. That's what's happening down inside here as the RNA is being built, and then finally when it reads, stop right there just a second. How does it know when to stop reading, class? There's a code that says stop. And then it stops reading. And by the way, there's another protein that helps it separate and leave. And then zip back together. Keep going. So the messenger RNA has now been made. What's not on this video, stop. What's not on this video is the whole maturing process and migration inside. You remember we talked about all that? That takes like what? 200 some odd different proteins. We're skipping all of that. So now here comes the messenger RNA outside the hole. You remember how many proteins it took to get through that hole? By the way, I forgot to tell you. Do you think you can go back through that hole? No, class. What do you think guards the hole going back? Exactly right. So you're keeping count, right? I've already told you we're over 2,000. All right, here we are outside, and there's the messenger RNA. Keep going, Jacob. And here comes the ribosome. Does not look like two ducks hugging each other. But there it is. See how it closed down? I stop right there just a second. Can you see the three-letter codes here that the messenger RNA has copied? That's the code for what? Amino acids, which makes proteins. And it's going to go through the messenger RNA, through the ribosome and be read. And now let's watch a little while. Go. He's going to go around now and open up the inside. But see these guys fluttering along here? Those are transfer RNAs that are bringing in the appropriate amino acids from your cytoplasm. Stop right there just a second. Folks, you better be eating appropriate amino acids because your body doesn't make them all. So you got to eat some of them. And they have to be floating around in your cytoplasm so the transfer RNA can pick them up and bring them in and make the protein. You with me, right? These are transfer RNAs. Let's go a little while longer here, Jacob. And I'll tell you when to stop. Get your finger ready. He does a nice job with this. Stop right there. This is a transfer RNA, which we haven't talked about, but it carries the specific amino acid. You see that one right there? Jacob, we're going to go forward a little bit. Stop. That's another one. Go ahead a little bit. Stop. That's another one. Go ahead a little bit. Stop. That's another one. There's another one. Keep going. Stop. There's another one. Keep going. Okay, stop. There are exactly, class, 20 transfer RNAs because there are 20 amino acids. Class, listen to me closely. Each of those transfer RNAs brings the appropriate amino acid up and says, here I am, here's the right one, matches the code, and then it adds it to the protein. In addition to that, each transfer RNA has a specific, what do you think? Protein to do the job or nothing happens. That's 20 more proteins. So when it comes up here and gets the job done, you have to have proteins helping it every step of the way. Look, folks, when people design a factory to build something, don't they have all kinds of jobs lined up that everybody does in order? Of course they do. This is better than anything men have ever built. Way better. You know how long it takes to build a protein 1,000 units long? Watch me. They just built 6 in the time it took me to do that. And you wonder why you get tired sitting there watching me? You got 30 trillion cells in your body. Every one of them's building proteins. You better stay healthy. All right, Jacob, keep going. We're going to try to go inside now and watch those transfer RNAs come in there and plant. Can y'all see okay back over here? Okay. See them coming in? There's one. He's trying to show it fast, but that's way too slow. Stop. My fault, Jacob. Keep going. Keeps going. We're going to go back inside in a minute, I hope. He's trying to make it faster. You see that? Stop right there. All right, here's the transfer RNA. Look, class, down here at the bottom is the code on the transfer RNA that matches this 3-letter code on the messenger RNA. But up here is the amino acid, which connects to the previous ones by a covalent bond to form the protein. The protein's up here and the code's down there. It's incredible. Folks, nobody could possibly believe, if you saw something like this in a human factory, that happened by a wonderful, slow process over millions of years by itself. No way. Keep going. Up the top here is the protein coming out. That particular one is hemoglobin, which is why he colored it deep red. You see how it's coming out one at a time? And then finally it comes to the terminator, and it lets it go down here, and it releases it into the cytoplasm. And you say, there, it's ready. No, it's not. This is post-translation. That protein, which is a string of amino acids, is folded up already. Can you see that? Because of the particular amino acids, it folds into a certain shape, but it's not finished. The next thing you have to do is make it fold more. Because if it doesn't fold precisely the way that protein has to be folded, it doesn't work and it gets eaten alive. And by the way, did you know part of the post-translation process is destroying proteins that have been damaged, killing them, and eating them? Not literally. But that's built in. Plus, if there's a protein, there was a mistake on it, there's a process built in that adds a little thing to it that says, take this one to the garbage. Built in. There have to be bunches of things done to this protein to get it ready to do what it's supposed to do. It's not finished. That's why you need 1,857 proteins to do this process. And when it finally gets finished, the books will say it is then released into the cytoplasm to do the job for which it was designed. And then they tell you, but it wasn't designed. And I say, Jacob, we need to go on. Can I just click? Yeah, no, I'm going back. Get me forward, Jacob. There we go. The chemistry of building proteins in every living cell is sufficient to proclaim there must be a great designer. Would you agree, class, with just what I've told you? But we have learned so much more about the chemistry of living things in the last 50 years. I haven't touched the hem of the garment today. I know you're worn out. You ain't heard nothing. The cell is a self-replicating nanoscale robot. It's not a glob of protoplasm like Darwin thought. He knew nothing about this. I believe if Darwin were standing here today and saw all of this, he'd say, I give up. His theory no more accounts for the existence of a cell with all its complexities. We call it self-replicating because a cell can and does reproduce every part of itself constantly. Don't miss tomorrow night. It's nanoscale because the cells are quite small. You know what a nanometer is, class? It's 1/billionth of a meter. So here's a meter. Divide that up into 1 billion equal parts. That's 1 nanometer. That's the kind of smallness we have here. And finally, it's a robot because its activities are carried out unconsciously and automatically by precision molecular machines that follow ordinary physical laws. What I have just shown you is just one little tiny piece of what's going on in every cell of every living thing. At an enormous rate of speed, without anybody in there being the straw boss. And I don't have time to talk about that, so we're going to skip to Anthony Flew. Professor of philosophy, atheist, author, and debater for 80 years. He was an avowed atheist, open, published lots of books. He was looked up to by the atheist community as one of their greatest heroes. In 2007, at the age of 80, he wrote a new book, There Is No, God. How the world's most notorious atheist changed his mind. I love a man like that. I hope I'm honest and humble enough. If I see in plain writing clearly that something's different than what I said, I hope I'm humble enough to say, I was wrong. So let's listen to him. This was in an interview. He said, it's become inordinately difficult even to begin to think about constructing a naturalistic theory of the evolution of that first reproducing organism. The enormous complexity by which the results were achieved looks to me like the work of intelligence. It now seems to me that the findings of more than 50 years of DNA research have provided materials for a new and enormously powerful argument to design. And I say, amen. And he got crucified by his atheist compatriots for daring to say something as common sense as that. I appreciate such a person. I wish for many more. Is it reasonable to believe in God in this scientific age? A lot of those guys that say, no, you're a mad, deluded lunatic. No, I think you are, if you don't believe it. It's more reasonable than it's ever been, class. I hope I stupefied you tonight. If not, please come back tomorrow and give me another chance. Because there's a whole lot more to this story. And God bless you for your good attention. You were patient. I'd give you an A, class, for attention, even if I did have to make you stand up once.